give a brief account on the mechanism of synaptic transmission, mechanism of vision, mechanism of hearing. So there are three mechanisms. So in a synaptic transmission, how does it work, how the vision works and the hearing. So let us learn about them. The first one is mechanism of synaptic transmission. That means the synapse which is present between a two neuron. So in the synapse, how the nerve impulse is being transmitted. So let us learn about it. So synapse is a junction between two neurons. So here we can see there are two neurons. The junction between the two neurons is called synapse. It is present between the axon terminal of one neuron and the dendrite of the next neuron, separated by a cleft, and this cleft is called synaptic cleft. So presynaptic terminal is the axon terminal and the postsynaptic terminal is the dendrite of the next neuron, and these two are separated by a space which is called synaptic cleft. So here also presynaptic terminal and postsynaptic terminal. Now the synapses are of two types. One is chemical synapse and the electrical synapse. In the chemical synapse, there is no connection between the two neurons. In the electrical synapse, the two neurons are connected by the gap junction channels. So let us see how the impulse will be transmitted in the chemical synapse and electrical synapse. Now coming to the chemical transmission. So once the action potential reaches the end of the axon, that is synaptic terminal, here the synaptic terminal contains vesicles which stores neurotransmitters. The neurotransmitters are acetylcholine. So these neurotransmitters are released into the synaptic cleft. So once the impulses reaches the end, the vesicles binds to the plasma membrane and the transmitters are released into synaptic cleft. Now on the dendrite of the next neuron, there are certain receptors and the channels. The receptors are present on the channel. So these receptors are the site onto which the neurotransmitter will bind. So acetylcholine diffuses into the cleft and binds to the receptor present on the membrane of the next neuron. So when it binds to the membrane of the next neuron, the ion channel opens and the calcium ions which are stored in the synaptic cleft enter into the dendrite of the next neuron. Inside, the dendrite of the next neuron becomes positively charged and it becomes depolarized. Now this depolarized initiates action potential in the next neuron. So this is how the information is being transferred in the chemical synapse. The electrical impulse will be converted into a chemical messenger called neurotransmitter, which initiates another electrical impulse in the next neuron. Now coming to the electrical transmission. It is a type of transmission. An electric current is formed in the neuron. Here, the electric current generates an action potential and leads to the transmission of nerve impulse across the nerve fiber. This is most faster method of nerve conduction when compared to chemical. So here we can see both the neurons are connected by a gap junction channels, and through this gap junction channels the action potential passes from one neuron to another, and therefore this is more faster when compared to the normal chemical synapse. So this kind of electrical synapse are less in the human body, more chemical synapses are present. So this kind we can see in the muscles, more not in the neurons. So moving to the next one, the mechanism of vision. Let us see how the mechanism of vision takes place. Now we have learned about the structure of the eye. So eye has got three layers, the sclera, choroid and the retina. Now this retina innermost layer has got three regions, ganglion region, that is layer of multipolar neuron, and then bipolar neuron, and then innermost side, that is the photoreceptors. So ganglion, bipolar and photoreceptors. So here we can see more clearly. So this is the ganglion layer, and at the center this region is the bipolar cells and this region is the photoreceptor region. And the photoreceptor region includes two types of cells, the rod cells and the cone cells. So these are the rods and cone cells which are arranged in the photoreceptor layer. Now, let us understand the mechanism of vision. Now, these photoreceptor cell is composed of a protein called opsin. Now, this is the protein called opsin. Along with this protein, there is aldehyde of vitamin A, which is also called as retinol. So, retinol and opsins are always present together in the photoreceptor cells. So, this is the rhodopsin, opsin and the retinol. When the light reaches on the retina through the cornea, so when the light enters from the outer layer to middle layer and to the inner layer, once it reaches the retina, it leads to the separation of the retinol and the opsin. So these retinol and opsin gets separated, and this separation changes the structure of the opsin. As they get separated, the structure of opsin gets altered. So when it gets altered, so the permeability of the membrane of opsin changes, and this creates a potential difference in the cell. So the potential difference is generated, that is the ions, the difference has been generated, and this leads to the formation of action potential. So we've learnt about how the nerve impulse is formed. So change in the potential difference causes action potential. So here also the action potential is caused due to the change in the membrane permeability of these rod cells. And this action potential will be passed on to the bipolar neurons. From the bipolar neurons, the action potential is passed to the ganglion neuron. So this action potential is passed to the optic nerve, and this optic nerve carries the information to the visual cortex present in the brain. The brain analyzes the information and stores the image. And this is how the image is formed in the retina. The image formed in the retina will be conveyed to the brain. So the mechanism of vision. So all this mechanism occurs in the retina. So retina, when the light falls on the retina, the opsin and the retinol structure changes. So this leads to the change in the permeability of opsin, generating an action potential in the opsin, which will be transported to bipolar neurons and then to the ganglions, and ganglions to the optic nerve, through which it is taken to the brain. Okay, the next part is the mechanism of hearing. So how the mechanism of hearing takes place. So the organ responsible is the ear. So ear has got three regions: the outer ear, middle ear and the inner ear. Now the outer ear includes pinna, auditory canal and the eardrum. So the pinna of the external ear collects the sound waves and it directs towards the eardrum through the external auditory canal. So this pinna collects the sound waves and through the auditory canal it hits the eardrum. And these waves strike the tympanic membrane, which is also called as eardrum, and the vibrations are created. And these vibrations are passed to the oval window present in the inner ear. So through the ear ossicles. So three ear ossicles are present: malleus, incus and stapes. The three bones present in the ear are called ear ossicles: malleus, incus and stapes. So this ossicles transmit the sound waves or the vibration to the internal ear. Now the internal ear consists of the cochlea. So this is the internal ear. When the sound waves reaches here, it reaches the cochlea, and this is transmitted to the fluid present in the cochlea, that is the perilymph. Yeah, this generates the sound waves in the lymph. So in this particular region. Now this region has got three canals: lower canal, middle canal and the upper canal. In the middle canal there is a membrane called basilar membrane, and on this basilar membrane the organ of Corti is present. On the upper canal there is a projection of layer called Resinus membrane, and this Resinus membrane have got tectorial membrane on it. Now on the organ of Corti there are specialized sensory cells called hair cells. So these sensory cells or hair cells are present on the organ of Corti, present on the basilar membrane. And the tectorial membrane is a projection from the upper canal. As the sound waves reaches the basilar membrane, the hair cells, that is the sensory cells, bends and touches to the tectorial membrane. When they touch to the tectorial membrane, the sound waves are converted into nerve impulse. That means there is a change in the membrane permeability and the action potential is generated over here. And this action potential has been transmitted to the sensory neurons, that is the auditory nerves. And through the auditory nerves, the impulse is taken to the brain. That is the hearing center of the brain or the auditory cortex of the brain. And this information is analyzed and the sound is recognized. So this is how the mechanism of hearing takes place. So students, here we learnt three different mechanisms: the mechanism of synaptic transmission, the mechanism of vision, and the mechanism of hearing.